talk about like the cell life cycle. We're going to talk about how the different phases of the cell life cycle and when things and what happens in those different phases. So most of you guys have seen the cell life cycle. It kind of looks like this, right? Now, what we get normally is that, right, most cells undergo this cell life cycle right here. With G1 phase, you just form that cell, it's going to go into a process called G1, S, G2. These are phases combined, we call the interphase. This sets up the kind of, the environment that eventually allows us to have mitosis. Once we have mitosis, then we can eventually split the cell in half, producing cells that are identical to each other, that are identical to the parent cell as well, right? So in the G1 phase, we're going to see that everything is growing. We're going to double the amount of nucleotides, double the amount of materials required to then enter G2, the S phase. The S phase is called the synthesis phase, where DNA replication happens, right? So again, we need to double everything. Even the DNA needs to be doubled. So in this S phase, cells replicate, all nuclear DNA replicates, even the centrioles replicate. So after the S phase right here, we should have double arm chromosomes instead of just one single arm chromosome from mom. Now have one double arm chromosome we call sister chromatids from mom. Now, once we split it, it's going to count for two though, right? So same thing for dad. Dad's going to normally have one single arm chromosome number one. So then you have chromosome number one copy from mom. Then you have chromosome number one copy from dad. That's perfect, right? All the way to 23 chromosomes, right? One copy of one of the 23 from each parent. So after we're done with the S phase, we double the amount of DNA. So then the cell has 92 chromosomes, two copies of the 46. But we don't call them separate chromosomes yet because each of those chromosomes are now linked together. And where they're linked together by that centromere, what it tells us is that, yes, they're linked together. They count only as one chromosome, even though there should be two, right? Now, once we undergo this synthesis phase, now we get ready to the G2 phase, where we double the amount of phospholipids, organelles, where we double the amount of just kind of everything. So then we can get ready to split, right? Even cell repairs DNA replication errors. They grow and synthesizes enzymes that control cell division. They increase the number of phospholipids and organelles. So then we are ready to divide, right? That division starts with the mitotic phase. In the mitotic phase, remember, we want to bring everything towards the middle. So then when we split everything, each cell, we're going to go from one cell to two. Each cell is going to have one copy of one chromosome from each parent. So you have two parents. So you're going to have one cell is going to have one copy of chromosome one from mom. One cell is going to have one copy of chromosome one from dad, right? That's going to be the way it goes all the way through to 23, right? So in the mitotic phase, we're going to have, right? And again, right? In mitosis, we're going to see that there are those phases, prophase, metaphase, anaphase, telophase, right? So kind of know what happens in each of the phases. Now, what is the function of mitosis? Mitosis is cell division where you're producing two genetically identical daughter cells that are identical to the parent cell, right? This is how we form one cell and eventually turn us into the 70 trillion, 80 trillion cells that make us who we are, right? So development of the individual from one fertilized egg to roughly 50 to 60 to 70 trillion cells that allows us to be who we are. Now, we also allows us to growth in all tissues and organs after birth. Remember, you can't add cells from the outside because they're not going to share the same genetic information. And that'd be like what would happen if you try to give somebody your kidney, right? And you don't type and match them, right? Though that kidney is going to be different. My kidney is going to have different genetic properties than anybody else's unless I have an identical twin, right? So in order to grow without any kind of rejection, we're going to have to grow from the inside, where one cell somehow reproduces itself to form two. Those two becomes four, all the way till we have 50 trillion, right? Mitosis also allows us to replace cells that die. Think about what happens when you have a sunburn and you have to replace those cells, right? Repair damaged tissues as well. When you get a cut, right? What happens? Well, that cut, we need to repair that damaged tissue, and that happens because all the cells below that cut from the stratum basale cells of the skin starts to hyper kind of go into mitosis very fast to replace all the damaged cells. So there are four phases to mitosis. In prophase, this is when we start seeing the genetic material. It condenses into compact chromosomes. Up until this point, we don't really see any chromosomes. Now we have 46 chromosomes, but they're double-armed sister chromatids per chromosome. When the nuclear envelope disintegrates, then centrioles sprout, right? And you can see these centrioles over here, they start sprouting. Now you start seeing the chromosomes a little bit better. The nuclear envelope starts to disappear, so then we can start moving the chromosomes around. In metaphase, right, what we're going to see is we're going to line all the chromosomes from one to another right down the middle. So here's chromosome number one from dad. Here's chromosome number one. That's a copy. So we have two chromosome number ones over here. Here's chromosome number one for mom. Here's a copy. Two of mom's chromosome number one. Chromosome number two from dad and a copy, right? Chromosome number two for mom and a copy. What we want to do is to split each of these chromosomes right down the middle right here, right down the middle. As we split it, you have one copy of chromosome number one from dad go to this side, and as it moves to this side, it'll go and become another cell. Then the other copy of the chromosome number one from dad will go to the other side and become part of a different cell. Now you have one copy of chromosome one from to this cell, one copy of chromosome one from dad to this cell. Perfect. That's what we want. One copy of one chromosome from each parent. So how about the other copy of chromosome one? Well, here's mom's copy of chromosome one. This one goes to this side. This one goes to the other side. So then now this side right here, which would be a different cell, has one copy of chromosome one from dad. The other copy of chromosome one is coming in from mom. Perfect. Same thing on this side. One copy of chromosome one from mom, from dad. The other copy, we split everything right down the middle, right? The other copy goes to, right, of chromosome one from mom. Chromosome two, we split right down the middle. So this arm comes to this side, right? One copy of dad's chromosome number two, right? Then the other copy of chromosome number two is from mom. One copy of one chromosome from each cell. Now, in metaphase, we just line everything up. In anaphase, we're going to split everything in two. So then each of the sister chromatids goes to the other side, right? Single-stranded daughter chromosomes now, because we're separated, they're no longer considered chromatids. They're now chromosomes. Each of the chromosomes will then migrate to the opposite side. Here's mom's copy of chromosome one. It's going to go to this side. It's going to turn a different cell here. The other copy, remember, DNA replication, we duplicated it. The other copy of chromosome one from mom goes to the other side. Chromosome one from dad, this side. The other copy, this side. Now, this side's going to have one copy of chromosome one from mom, one copy of chromosome one from dad. Perfect. One copy of chromosome one from mom, one copy of chromosome one from dad. Perfect. This is why we line it up like this. So when we split it, each of the newly forming cell will have one copy of one chromosome from one parent, right? The other copy will be from the other parent, right? The splitting occurs in the anaphase. And then as we move it to the sides right here, we have microfilaments that kind of pull it to each side, right? As we pull it to each side, then right down the middle, we have what's called a cleavage furrow. And then the cleavage furrow, that's where the split is between the cytoplasm, right? So then we can form two distinct different cells. In telophase right here, right? Telophase chromosomes cluster on each side. Rough ER makes a new nuclear envelope around each cluster. Chromosomes uncoil to chromatins. The mitotic spindle disintegrates. By the time we get to telophase, we've already done all the hard work. We've already split the chromosomes. So then each cell has different, each cell has one copy of one chromosome from one parent. And, right, now we're ready to split the cell in half, right? The last part, cytokinesis, that's where we completely separate the cells. Up until then, right, they're still stuck together in telophase. At the end of telophase, cytokinesis occurs where we pull the cells apart, right? And that's it.